Couri from Cleveland Clinic.
Good morning. I want to thank the committee for the opportunity of presenting my work. The title of my work is Pelvic Organ Disorder in Lysyl Oxidase Like 1 Knockout Mouse Model and Innovative Preventive Therapeutic. I have no conflict of interest to disclose, and I want to thank the ICS committee for the travel award. Two of the major risk factors for pelvic organ prolapse are pregnancy and delivery. There is a diffuse damage of muscle, nerves, and connective tissue, especially with a prolonged second phase of labor. This damage will progress over years until prolapse occurs. Nowadays, no treatment is preventive nor target the pathophysiology of the disease, and many patients will undergo surgery for treatment. The ideal treatment should meet this criteria, should be delivered before significant anatomical defects, should localize the areas of injury, and should facilitate repair of muscle, nerve, and connective tissue. And stem cell-based therapy has the potential for this. For this study, we used bone marrow-derived stem cell. This is a cell lineage that is well studied and commonly used in models of injury. It is immune privileged and can be used in an allogenic fashion without major immune reaction. These cells secrete growth factors and cytokines that can act systemically. These secreted factors can be collected in the media culture and concentrated to be used as a treatment that can be used and act systemically. The control is produced on the same fashion without being conditioned with cells. To study this, we cannot start on a clinical trial. So an animal model is important. I wish having mice volunteering to deliver samples like that will be this easy. So to do this study, we used the Laksa 1 knockout mouse. This is an animal model that is deficient in the enzyme Laksa 1, which is important for the synthesis of elastin. It simulates the prolapse in women. It has defective connective tissue and similar prolapse phenotype. Pregnancy and vaginal delivery are necessary. It is delayed, but not totally prevented by C-section. It will progress with age, and it's correlated to stress urinary incontinence. And with this animal model, we can also use a grading system similar to the pelvic organ prolapse grading system, the POPQ. We call the MOPQ. It's the mouse organ prolapse grading system. Here you can see on figure A and B, animals that are not prolapse. The prolapse, it's not the rectal mucosal protrusion. It is the bulging that is presented by the black arrow. So we consider grade 2 and 3 as prolapsed animals. The hypothesis was that the CCM, the concentrated media from the MSCs of wild-type animals, positive CCM, would have a greater protective effect against development of prolapse than CCM from Laksa 1 knockout, the negative CCM. The aims of this study was to assess relevant protein content of CCM from Laksa 1 knockout animals and wild types, and also to determine if intraperitoneal IP injection of either CCM has a protective effect against development of prolapse. So to do this, we harvest bone marrows from these animals. We culture and expanded the cells, the MSCs, from both the knockout and the wild-type male animals. We cultured the cells and expanded until passage 8 to 11 in medium with serum. These cells were cultured after reaching the passage that we were looking for. We cultured for 24 hours in a serum-free medium. The cells were discarded and we got only the media and concentrated 50 times. We used a control, just the serum-free media that was incubated for 24 hours on the same way, but they were not conditioned with cells. To do the CM, the control, and the CCM analysis, we used Bradford protein assay for total protein concentration and ELISA for individual protein concentration of relevant proteins for connective tissue. The animal model study design was done using 8 weeks old Laksa-1 knockout females. They deliver after around 21 days after being pregnant, and they were randomized in 3 different groups: control, that were given CM, negative CCM, and positive CCM. After second delivery, these animals were kept on the same group and received the same treatment as they received after first delivery. And we did the MOPQ assessment weekly, starting the first week after first delivery. Here you can see on the y-axis the percentage of animals that didn't prolapse. This is the Kaplan-Meier curve. And at the x-axis, you can see the number of weeks after first delivery. We could see a significantly difference between animals that were treated with negative CCM compared to the controls. And we couldn't see the significant difference when we compared the positive CCM treated animals to the controls. There was no significant difference comparing negative CCM to positive CCM treated animals. The total protein concentration showed that under control, as we expected, they were not conditioned with cells, there were no protein detectable. And there was no significant difference in the amount of, on the concentration of the negative and positive CCM. The specific proteins relevant to connective tissue metabolism, we could see that Laksa-1 were not detectable from the knockout animals, and it was on the wild type. Assessing LOX, CCL7, CXL12, VEGF, TGF-beta, FGF, Activin A, TIM2, and MP2, there was no significant difference when we compared positive CCM and negative CCM in any of these proteins. In conclusion, we found that IP injection of negative CCM, which lacks of Laksa-1 protein, slow development of pelvic organ prolapse. CCM slows development of prolapse via mechanism other than replacing the missing Laksa-1 protein. Non-invasive cell-based therapy could be potentially used, possibly in an autologous fashion, to prevent or delay development of prolapse. Thank you.
Any question from our audience? No, I have one question. Congratulations for a nice paper.
Thank you.
I'd like to know if, as the urinary incontinence, published in the recently of the paper using cell culture to treat urinary incontinence, do you believe to answer us is possible to treat POP using cell culture now? Or if not, what is the next step to provide this?
Well, we hope to use not only cells, but the media secreted from these cells, the protein secreted from these cells that will generate less reaction. And that's what we are trying to prove here, is to increase the number of these animals and find what is secreted on this media, what are the differences that we couldn't find so far to prevent the prolapse, the pelvic organ prolapse on these animals.
Okay.
Cara Tanimum from Montreal. Thank you very much for the presentation of what sounds like some very challenging research. I was curious why you decided to harvest the cells from male mice. This is a female-specific condition. Is there any reason, and you're talking about the future doing autologous stem cell transplantation, why would you select male mice for this experiment?
There's no specific reason why we decided to use. It's just because it's easier, because we use the females to inject, and it's just easier to use the males. That not as useful to harvest the bone marrow.
Would it have been different if they were female mice?
Sorry?
Is it possible that the results would be different if you harvested from female mice?
So that's a next step of our study, to do the analysis of the CCM and the cell culture from the females to see if there's like any hormone interaction or factor related to that. That's a good question. That's what we are working on right now. Thank you.
Hi. Nice presentation. Congratulations. I'm Maria Bertolini from Brazil. So I have one question, and it is like, why you decided to inject intraperitoneal, the CCM, instead of locally on the pelvic floor per se? And also, I'd like you to comment something that you about your model of prolapse developed in knockout mice. Have you tried like to treat other kind of models of prolapse?
So the first question was just, sorry, the first question.
Why you not inject your CCM locally?
Because you have like a diffuse damage of muscle connective tissue. It's really hard to target one single point. We cannot inject the entire vagina. It's really hard. On the other hand, like for stress urinary incontinence, we assume that it's like easier. So that's why I use intraperitoneal injection, because it's abdominal and connects systemically. And no, we didn't use this treatment on any other different animal model. We work exclusively with this animal model.
Okay, thank you.
Okay, good job. Congratulations. Move on for the next
Thank you.
article.